All right, everybody, I'm going to go ahead and go through a little bit of a review for the test that's going to be happening tomorrow. You can see right here I just have your notes packet, and I'm going to kind of talk my way through this a little bit as a good review. So first off, you can see standard 1, 2, 3, and 4. I'd make sure you look through those and that you can identify the components and chemical properties of blood. What are these items? What are they called? What's the scientific name? What do they look like? Okay. Same thing with genetic probabilities. You know how to do a Punnett square. Next one is looking at different types of blood spatter. No, low, medium, and high velocity impact spatter. And then different types of drip patterns, cast off. I mean, you could read through these. And I feel if you did your flashcards, you know these. Okay. And then standard 4 is those presumptive blood tests. Okay. All right, so you can see there's the standards. This vocab you should feel comfortable with. I know the vocab term that we did not talk about was surface tension a whole lot. That was way back in first trimester. So just a reminder that blood holds together in a nice little circle droplet because it has water properties, adhesion and cohesion, right? So it's a cohesive water sticking to water, okay, and creating that surface tension that allows water droplets and blood droplets to act the way that they do. If there's any of these terms that you're uncomfortable with, make sure you look through your notes packet or look it up online, or you can message me. All right, you can work through this little crossword. Once again, if you get caught up on anything, make sure you send me an email, a remind message, whatever is convenient, and I can help you. All right, so here's the first page. Just knowing the anatomy of your blood, right? So you should know the scientific name for your red blood cells, your erythrocytes. And you look at your erythrocytes, leukocytes, and your thrombocytes, okay? Those are your 3 types of cells. Well, which one is the biggest? Okay, you should know that your leukocytes is the biggest. That's your white blood cells fighting off infection, producing antibodies. The second one's your red blood cells, and that is huge when it comes to your presumptive blood tests in forensics, because that's what your luminal and your hydrogen peroxide and your Kastle-Meyer reacts to, is the hemoglobin found on those red blood cells. And then third, your thrombocytes, also known as your platelets. Those are your clotting cells. So biggest to smallest, white blood cells, red blood cells, platelets. Which one do you have the most of? Definitely your red blood cells you have the most of. Okay. Now, what percent of your blood is made up of plasma? 55% of your blood is plasma. And 90% of that is water. So a large portion of your blood is made up of water, which is why we know that blood acts like water. It has those water properties, those cohesion, surface tension. All right, we get into blood typing. And if you have A blood, you have A antigens. If you have B blood, you have B antigens. If you have AB blood, you have both A and B antigens. And if you have O blood, you have nothing. So then you got to think about your antibodies. Okay, you always make the opposite of what you have. So if I have A antigens, I'm going to make B antibodies. If I have B blood, that means I have B antigens and I make A antibodies. So you make the opposite of what you have. Now, AB blood is a universal receiver because it doesn't make any antibodies. And then O blood is a universal donor because it doesn't have any A or B antigens on it. And so if you donate O blood to somebody else, the body's not going to react in a negative way because there's no proteins on it. It's going to freak out about because there's no A or B antigens, those proteins on the red blood cell surface area. Okay, then of course you kind of get into the Rh factor, and that Rh factor, kind of scroll down here a little bit, right? This Rh factor just means if you are positive, you have the Rh protein, and if you're negative, you don't, right? So can somebody that is positive donate to somebody that is negative? No, because it has the Rh antigen. Their body will freak out and make the antibodies, and then agglutination is going to happen and it's going to clump. So positive can only donate to positive people. Negative can donate to everybody. All right, genetic vocab. Little review, genotype is what your genetic makeup is what your alleles are. And phenotype is your physical outward appearance. So your phenotype can be misleading because whatever your phenotype is doesn't mean you don't carry a secret hidden allele in your genotype that you don't know about. And then all of a sudden it shows up in your offspring. So if you're trying to figure out what your kids are going to have, you care about your genotype. Okay, homozygous dominant, homozygous recessive means they have two of the same alleles. Dominant means they're both dominant, recessive they're both recessive. Heterozygous is a hybrid, it's a mix. You have a dominant and a recessive allele. Okay, here's your Punnett squares. There's the practice problems. You can kind of review through those. We talked about how O blood is by far the most common blood out there, and AB blood is the least common blood out there. So in this case, you guys that are, well, hopefully not, but if you're going to commit a crime, you want to hope to have O blood because you have the largest class group of people that also have the same blood type as you. All right, rolling on. Okay, this is what we started third trimester, is getting into all this blood spatter. Okay, first off, if you're going to be a blood stain pattern analysis individual, there are three things you got to make sure you focus on. And it is the shape, the size, the distribution of your blood. Okay, and that is the shape relates to its angle of impact. If it's more circular, it's 90 degree, more elliptical, elongated, it hit at an angle less than 90 degrees, right? The size affects the velocity. The bigger the drop, the slower it was moving, and the smaller the blood droplet, the faster it was moving. And then the distribution. That is helping you to go through the blood spatter and what happened, right? Was it cast off? Was it missing? All right, so this is kind of what we just talked about, the size of the blood spatter, the shape, directionality is huge. Directionality tells you where did the blood come from. So you'll want to review through all the different types of blood spatter, and I'm going to show you some pictures here in a minute. But until then, you got to remember low velocity impact spatter, it's only going to travel up to 5 feet, right? And the blood drop's going to be larger than 3 millimeters. But if it's medium velocity impact spatter, the blood drop's between 1 and 3 millimeters, and it can travel 25 feet. And then finally, if it's high velocity impact spatter, less than 1 millimeter, and it can travel up to 100 feet per second. So that one is definitely going to travel some serious distance. Okay. All right, so 3 categories of blood. You have passive, and that means that gravity acted alone to cause it to fall. That's it. That's the only force that was acted upon it was gravity. You have projected, and that means gravity and another external force acted upon it. So let's move on down here. Here is our passive, and then here's our projected. So an extra force acted upon it besides gravity. And then finally, you got your transfer contact stains. And transfer contact is blood from a primary surface landed on a secondary surface where there wasn't any blood. Right? Hopefully you were able to go back, look through your notes of which ones fall into what categories, but we'll do that here in a second. Let me pull this on up. Okay, we've seen this before. So first off, I would make sure you look through this and know what category and what type of blood spatter is it. Okay. All right, so first off, A, this one is projected, and it is cast off, which falls under medium velocity impact spatter. All right, B, this is passive, and it's drip pattern. It's blood dripping into blood. And because of that, you get all this satellite spatter. You get the parent drop, and then if we zoomed in on that parent drop, I'm sure we'll be able to see that there are spines. Okay, C, this is projected. It's an arterial spurting, and typically that also falls under medium velocity impact spatter. All right, D, we can kind of zoom in here a little bit more, can't we? D, this is your high velocity impact spatter. These blood drops are less than 1 millimeter. They traveled up to 100 feet. All right, E, this is an example of low velocity impact spatter. This blood droplet is larger than 3 millimeters. Once again, you see good spines. You can even see some satellite spatter. F is passive, and these are just passive blood drops. 90-degree angle, nice circular shape. You see the spines, and some itty bitty little satellite spatter. All right, G is projected. Same with E and D, they're all projected. And this is medium velocity impact spatter. And these blood drops are going to be between 1 and 3 millimeters, and once again, traveled up to 25 feet. I like this image because you can really see, let's zoom in a little bit more, you can really see the directionality. Okay, you can see where the tail, so it came from this way and it's traveling this direction, and then gravity is pulling those drops down, so you're getting some blood flow here. All right, H, this is transfer, and it is a smear, also known as a swipe. I is also a transfer contact stain. Now J, it is projected. Okay, this is expirated, coughing out blood, filling up of your lungs, your chest, your throat region. And K, there's two things going on here with K. You have passive blood drops, but then a transfer contact happened and it got wiped through. So here is your wipe pattern. And then finally, you do have these skeletonized blood drops where it was drying and then it got wiped through. So you got skeletonized. All right, so that kind of reviews through the different types of blood spatters we've talked about. All right, now directionality. Okay, this is so important when it comes to determining the point of convergence. Okay, point of convergence, you need two things: angle of impact, directionality. Once you have those two items, then you can use that to plug and chug into the formula to figure out, well, your angle of impact, you plug and chug that into the formula to determine that degree of which it came off the wall. And then you can put all those together to figure out where did they converge and merge together to see where that person was standing when the blow hit. All right, moving on down. You got some blood facts. You can review through that. The big thing I want to look here now is the secretor. If you are a secretor, you can use your saliva, your tears, other bodily fluids to determine what your blood type is. A, B, O, or AB blood type. Okay, finally get to our presumptive blood test. Okay, so Kastle-Meyer, this is the one that I think is kind of boring. It's a 3-step process. First, you put on a little alcohol and that cleans it. Then second of all, you put your phenolphthalein on, and that's your pH indicator. And that's what's going to turn pink in the presence of oxygen. Finally, you add that hydrogen peroxide. Okay, now the hemoglobin is going to chop up that hydrogen peroxide and release those oxygens. Phenolphthalein is going to grab those oxygens, and that's what causes it to turn pink. Some weaknesses is there are false positives, and it's going to turn pink no matter what after a duration of time. Strength's really sensitive. You've got this where it's 1 to 10,000, 1 drop of blood to 10,000 drops of water. You can still detect it. Now, hydrogen peroxide, it turns a white foam, and same thing. It's the hemoglobin, the iron in the blood, that breaks apart the hydrogen peroxide, releasing water and oxygen. Okay, luminol, this one, strengths, it's more sensitive. It's 1 drop of blood to 1,000,000 parts per water. It is more expensive, but it's very, very, very sensitive. And it can last up to a minute as far as the duration of the glow. Okay? Weaknesses, lots of false positives. We've got metals. We've got, you can't distinguish, any of these blood tests can't distinguish between animal and human blood. Okay? This is the luminol's the one that's got the horseradish, is one of the false positives. Copper, once again, different metals. Bleach can create false positives. So that's why it's presumptive blood tests, because it can create this idea that it is blood. Then they take it back to the lab and actually it isn't. Okay. All right, this is the extra credit. Okay. Looking at it, making sure when you collect a blood stain, it always has to be dried first, and it has to be packaged in a porous material. You care more about that than anything. Phlebotomist is that you draw someone's blood intravenously. Forensic serology DNA criminalists, these guys are only in the lab analyzing blood and fluids, but a serology technician collects the blood in the crime scene and then goes back to the lab and analyzes it. All right, that is, in a nutshell, 15 minutes of the entire notes packet. Okay, hopefully this helps you get ready for the test tomorrow. All right, good luck.